Good morning. My name is Ariel Furst, and I'm an assistant professor in the Chemical Engineering Department at MIT. And today I'm excited to tell you about some work from our lab on sensing bacteria and bacterial sensing. I'm sorry I won't be able to be there for the question and answer session, so please feel free to reach out to me by email shown here or on Twitter. Before I get into the specific projects we want to discuss today, I wanted to go over conventional electrochemical sensors and kind of how they work. So I'm sure all of you are familiar with the gold standard of electrochemical sensors, the glucose meter. It's shown on the bottom here. In a conventional sensor, you have a target or an analyte molecule. It binds to a biorecognition element, such as an antibody or a DNA aptamer on an electrode surface. And this binding event changes the surface properties, allowing us to transduce a signal and convert it into an electrochemical signal. These sensors have very high specificity and sensitivity. They can even go sub-picomolar, and they allow for very rapid readout with inexpensive and portable instrumentation. But what my group has continually realized is that just monitoring concentrations of individual compounds or biomarkers is insufficient to determine if there's contamination or illness. So what we work on is functional assays that are instead based on some sort of biological activity of either the target or the recognition element to improve on current features of electrochemical biosensors. With functional biosensing in mind, today I'm going to tell you two stories about how we can use the inherent properties of bacteria to both detect pathogens and detect small molecule contaminants in the environment. And the first story I'm going to discuss today is bacterial pathogens. Specifically, we're interested in looking at E. coli. They're a component of our native guts, and most infections caused by E. coli are actually endogenous. So that just means that E. coli from our bodies migrate to places where they shouldn't be. And although many different strains exist, they have varying virulence. And the most common ailments caused by E. coli are gastroenteritis and urinary tract infections. So E. coli can serve as both urinary tract infection pathogens as well as foodborne pathogens. Foodborne pathogens cause nearly 50 million illnesses annually and over 120,000 hospitalizations. And one of the challenges when we think about monitoring E. coli contaminants is when we think about food production, we need to be able to monitor from farm to fork. What this means is we need a sensor that's easy enough to use at production or in the farms themselves, in processing plants, in retail stores such as restaurants and grocery stores, and even in your own home. Similarly, when we think about E. coli as a urinary tract infection, it's one of the most prevalent infections in the United States. Nearly 100 million people acquire a UTI each year, with 25% being recurring infections. Because of these recurring infections, about 90% of UTIs have at least one antibiotic resistance. And because of the cost and time required to currently diagnose a UTI, many physicians prescribe antibiotics without even testing patient samples. So what we've done is we've developed a rapid and easy to use electrochemical assay to monitor and quantify pathogenic E. coli from both food and urine. The overview of this sensor involves feeding E. coli that might be in a sample a synthetic amino acid that they can incorporate into their surface. This then reacts with the molecule that we've modified an electrode with, and we can then detect these bacteria with high sensitivity using impedance electrochemistry. I also want to point out on the right that these sensors were developed with disposable electrodes that are very inexpensive and easy to use and require only 10 microliters of sample. So the way this works is we've synthesized this boronic acid containing synthetic amino acid. You can see that on the right. We can feed that to E. coli, and if those E. coli are alive and viable, they will incorporate that through peptidoglycan remodeling into their surface. This boronic acid head group will then react with a carbazide that's displayed on our electrode surface, which we've synthesized as shown on the right, that contains a terminal carbazide at one end and a thiol at the other to attach to gold electrodes. It's important to note that the E. coli have to be alive in order to incorporate this amino acid. So only those virulent bacteria are going to be monitored with this assay. So we could image our electrodes. Shown here are just 3 different images by scanning electron microscopy of our gold electrodes. The pebbly looking areas are different regimes from these screen printed electrodes. And we can see if we have the bare electrode and boronic acid modified E. coli, we see no binding to our surface. If we have our surface modified with that carbazide diol and E. coli that weren't fed that synthetic amino acid, we see no binding. It's only when we combine that carbazide electrode with the boronic acid modified E. coli, as shown on the far right, that we see cells present on the electrode. However, SEM is obviously not an ideal method to monitor for point-of-use systems. So what we did instead was use electrochemical impedance spectroscopy. Fundamentally, this is just resistance to AC current. And the way it works is you can divide your impedance signal into two different regimes, an electrolyte or diffusive resistance regime, which is basically from diffusion of these redox active molecules, and an interfacial or charge transfer resistance regime, where you're looking at what's happening at the surface of your electrode. What we're interested in is this charge transfer resistance, because basically that tells us how much of our surface is blocked to these small molecules. And from there, we can model this system as an equivalent circuit shown on the top, and then display the data as a Nyquist plot shown on the bottom. And what I want you to take away from this is simply that if we look at this arc on the bottom, the diameter of that is our charge transfer resistance. And so what we would expect to happen is if we have no cells bound, our charge transfer resistance would be fairly small. As the E. coli bind to our electrode and block access of that ferrocyanide and ferrocyanide, we anticipate that that charge transfer resistance will increase, as shown on the right, based on the number of cells bound. And that is exactly what we see when we implement this sensing method with our device. So shown on the top here is just a reminder of our process. We take E. coli that might be present in a sample, and we incubate them with the synthetic amino acid. If the E. coli incorporate that amino acid, they will be tethered to our electrode surface through a covalent bond. We can see on the bottom, we have our raw impedance data in these dotted lines, and our curve fits based on our equivalent circuits in these solid lines. We can see on the bottom that if we have no cells present, we see a very low charge transfer resistance in black. And if we don't add any boronic acid to the cells, we similarly see very low resistance. Similarly, if we add an off-target microbe, in this case Staphylococcus aureus, even at very high concentrations, 10 to the 7th CFU per mil, we see no response. It's only when we add those E. coli modified with that boronic acid, even down to 100 cells per mil, shown in blue, that we see this increase in charge transfer resistance. We can actually quantify this. So shown in the purple is just our E. coli titrated in buffer onto our electrode. And we can see that we have a limit of detection of 12 CFU per mil. And we have a linear range of detection all the way up to 10 to the 7th CFU per mil. Similarly, we have similar sensitivity for E. coli and synthetic urine shown in blue. Shown here is the titration of those cells with that boronic acid added into synthetic urine, and we can see that we maintain our sensitivity. In teal, we see a slight decrease in sensitivity when we attempt to detect E. coli from milk. We anticipate this being due to the large number of proteins that can add to our surface because of their presence in the milk system. But what we do see similarly is that for Staph aureus and E. coli that weren't modified with the boronic acid, we see no response. So although we do lose some sensitivity in very complex mixtures, we maintain an LOD at very low limits. This is 2 orders of magnitude below the infectious dose of E. coli. And we can detect E. coli specifically in lieu of Staph aureus being present. And so now we wanted to test this with real samples. So conventionally, you can see on the left at the bottom, we have agar plating with colony counting. This is literally when you plate a sample on an agar plate and just allow the cells to grow. Each colony indicates 1 CFU present in your sample that was plated. This is highly error prone and highly susceptible to slight changes in the protocol that's used to actually do the assay. So what we did here is we acquired eggs, romaine lettuce, spinach, raw chicken, all from a local farmer's market, and we also acquired a feline urine sample that was from a vet that had confirmed that animal had a urinary tract infection that was E. coli. We can see here in purple quantification of the cells present by our electrochemical measurement, and in gray is our CFU counting. So importantly, in all cases, we see that our electrochemical measurement gives us on the same order of magnitude as our colony counting, but we have significantly lower error consistently with our electrochemical measurement. And so what this has allowed us to do is to detect endogenous pathogenic E. coli from these complex mixtures. And so in summary, we've been able to develop this easy and disposable platform that allows us to both capture and detect pathogenic E. coli. Our sensitivity is 12 CFU per mil in both buffer and urine, which is orders of magnitude below the infectious dose. We only sense viable cells because peptidoglycan remodeling is necessary for the incorporation of that synthetic amino acid. And finally, we can detect endogenous E. coli in both food and urine samples faster and with less inherent error than colony counting. So this is how we can use the inherent activity of cells, in this case peptidoglycan remodeling, to detect pathogenic bacteria. But what we also wanted to do is to think about if we could use beneficial properties of cells to detect chemical contaminants. And specifically, what we were interested in detecting are organophosphate pesticides. This is a class of chemical pesticides that makes up about 50% of the pesticides used globally. Importantly, these chemicals have the same mechanism of action as chemical warfare agents. And it's estimated that there are about 385 million annual unintentional poisonings, resulting in nearly 11,000 deaths because of access to these chemicals. Not only is acute exposure a problem, but prolonged exposure is known to cause respiratory illnesses, diseases, cancer, and birth defects. And so what two students in my lab, well, postdoc Amrita Karbakar and an undergraduate, Rachel Almark, did was they developed a microbial mixture in which one cell, in this case E. coli, was engineered to degrade those pesticides. And then we engineered electroactive microbes to electrochemically detect the degradation products. So we'll go through this in a little more granularity. So when we look at organophosphates, the pesticides are shown in the middle here. The main two classes are paraoxin and parathion, and they're all connected. They all have this connection between a nitrophenol group and a phosphate group through that oxygen. We can see that this organic linkage through this oxygen is very similar to the structure of sarin, somin, and cyclosarin gases, which are chemical warfare agents. But the advantage we have in detecting these pesticides is that they all enzymatically degrade to the same compound shown on the bottom, p-nitrophenol, or PNP. And so what we can do is we engineered E. coli to surface express an enzyme, organophosphate hydrolase, that can degrade these pesticides. So it takes paraoxin and parathion and converts it to PNP. This PNP product does have a yellow color and currently is detected by UV vis. But the problem with this is that there are many chemicals in nature that are very similar in structure to this, and the sensitivity of UV vis is very low. It's actually high micromolar, which is insufficient for real-world detection. But we can see here that using these cells, we can degrade paraoxin to PNP as monitored by UV vis on the y-axis. And as we increase the concentration of the initial paraoxin, we increase the concentration of PNP, showing that our cells are catalytic and not the limiting factor here. Similarly, we can change the rate of this degradation based simply on varying the amount of cells we use. I also do want to note that in both of these cases, the cells are actually freeze-dried. So we are able to take those E. coli, surface express this protein, and then freeze-dry those cells and maintain that enzymatic activity. So we don't have to keep these cells alive in this assay. But as you could see on the last slide, the sensitivity of this assay was actually not that great. It's about 5 micromolar at best using UV vis. So we wanted to see if we could engineer in a circuit in cells that allows us to more sensitively detect these chemicals. So we found a transcription factor, DMPR, that when it's in the presence of PNP, is activated and turns on gene expression. So the first gene we put under its control was GFP, or green fluorescent protein. We can see just below that in the presence of that small molecule degradation product, we see a significant amount of fluorescence. While without that degradation product, we see little to no background fluorescence. So this indicated to us that this circuit would work for detection of this degradation product. We can see on the right that we were able to put this circuit into cells, in this case Shionella onadensis, and as we looked at GFP fluorescence, we can see that it's specific to this PNP. If we look at it with paraoxin, the starting pesticide, or if we take wild-type non-engineered cells and look at them with this PNP, we see no fluorescence. The problem with fluorescence is it's very difficult to get quantitative readout without hundreds of thousands of dollars of equipment. And one of the advantages of Shionella onadensis is that they're actually electroactive microbes. So these cells are facultative anaerobes, which means that they can respire oxygen, but in the absence of oxygen, they can respire extracellularly onto metal oxides present in their environment. So this means that they can reduce these insoluble metal oxides for respiration. And the way they do this is because they contain a series of cytochromes in their outer membranes. And these cytochromes act as a wire, allowing electrons to flow across the insulating membrane from the inside of the cell to the outside. We can see in the middle here that this is driven by a set of 5 different cytochrome-containing proteins. SYM-A is in the inner membrane. MTR-A, B, C, and OMCA are near or on the outer membrane. We can see here on the right that when we have these bacteria that colonize a carbon electrode, we see current grow in slowly over time over the course of several days. So we wanted to take that same DMPR transcriptional regulator and see if we could put it under control of SYM-A so that SYM-A is now expressed instead of that green fluorescent protein. So what we would expect to see here is that in the absence of that small molecule, we would see no current. And as we add that small molecule, it will turn on SYM-A expression and we'll see current grow in. And we can see on the bottom here the simplicity of these electrochemical bioreactors with cells. They're very small kind of mason jars. They're only about 50 mils, and they contain a very inexpensive carbon cloth electrode. This is a 3-electrode system, so we also have a reference electrode that's silver chloride, as well as a counter electrode. We can see the photo on the right next to it, this whole device hooked up. And what we can see on the far right in the graph is that if we have those Shionella that were engineered to have the circuit in them, plus 20 micromolar of the small molecule, we see that current grow in with a very strong signal. We see a very small amount of current when we have these engineered cells with 20 micromolar of the pesticide, and we anticipate that this is due to a small amount of the pesticide actually naturally degrading in the presence of UV light to that PNP product. And if we have either those cells only in light turquoise or 20 micromolar of the small molecule only, we see no response. So that signal is specific to this gene circuit being activated. And so what we sought to do next was what poses a significant challenge for much of synthetic biology, which is to combine multiple different species of cells into a stable co-culture. Here, though, we have the advantage that the E. coli don't actually have to be alive and viable in order for them to degrade this paraoxin. So the fact that we can use these E. coli that surface express this organophosphate hydrolase enzyme in a lyophilized form basically takes away much of the challenge with co-culture systems. So what we wanted to see now is if we start with that pesticide, that paraoxin, could that lyophilized E. coli degrade that to PNP, which is then fed to those engineered Shionella to generate current? And the answer is yes. Now we're looking at total charge, which is basically the integration of those current signals I showed before. You can see on the far left here that if we have both cell types plus the pesticide, we see a significant amount of charge accumulate. If we have just the cells and the paraoxin, just the Shewanella and the paraoxin, we see a small amount of current and charge. And this is, again, likely due to a little bit of background degradation of that paraoxin to PNP. If we just have the co-culture of cells, we see no background current. And similarly, if we have only the pesticide, we similarly see no charge. What was really surprising to us, though, is what's shown on the right, in that this sensor was actually quantitative. So not only do we see this increase in charge with increasing concentrations of that pesticide, but those charges are proportional to the concentration of pesticide present. So you can see here that we get a very large charge accumulation with 20 micromolar of the initial pesticide. We similarly see about half of that amount of charge if we use 10 micromolar, and it's halved again to 5 micromolar. And we do maintain signal above the noise, even down below 1 micromolar. So this means that our limit of detection here is sub-micromolar. And what that further means is that we have several orders of magnitude higher sensitivity than conventional UV vis to monitor PNP. Additionally, this system has given us a single device that allows us to both degrade these harmful compounds and detect and quantify their degradation products. Importantly, though, there are many similar molecules in nature to p-nitrophenol. So we needed to test and make sure that not only was our sensor working for other pesticides that have similar structure, but also that there was no interference from native small molecules. So we can see on the left here that if we evaluate pesticides that are similar in structure to paraoxin, including paraoxin, methyl, and parathion, we similarly see charge accumulate, as you would expect, because these degrade the same PNP product. In contrast, if we use a less prevalent pesticide, maloxin, that does not degrade to PNP, we don't see a response from our sensors. And this gives us confidence that we're specifically detecting this class of pesticides that degrades to PNP. As I mentioned, there are other small molecules that are similar in structure to PNP in nature. So we also tested several of these to make sure that they don't interfere with our transcriptional regulator. So here we have p-nitrophenol, as well as dopamine, hydroquinone, and 4-methylcatechol. And as we can see here, the only response, in this case we're back with the fluorescent strains, the only response we see is to p-nitrophenol. We don't see any response with these other small molecules that may be present in our samples. And so in summary, what we've been able to do is to combine the inherent ability to engineer E. coli with genetic manipulation of these Shewanella onadensis that are electroactive microbes to both degrade harmful environmental contaminants and detect the degradation products. When we combine these together, we can take these organophosphate pesticides that cause thousands of illnesses and deaths annually and degrade the pesticide and quantitatively detect those degradation products. Further, our limit of detection with these is sub-micromolar levels, which means we outperform both colorimetric and fluorescent sensors by several orders of magnitude with much less costly setups and equipment. And so, what I hope you've taken away from my presentation today is the importance of using and relying on the inherent properties of the biologicals you're incorporating into your sensors in order to improve their sensitivity and specificity. As we saw in the first example, we can ensure that we're only sensing these viable pathogens by specifically monitoring their binding through the incorporation of a synthetic amino acid. In the second example, we looked at how we can improve upon conventional sensing technologies by incorporating very precise biological circuits along with dually engineered strains. And so what we've been able to do here is to improve both the sensing specificity and sensitivity while also increasing the ease of use and decreasing the cost of devices to detect both these harmful bacteria and this harmful class of chemicals. So with that, I'd like to thank my group. They are really an incredible group of students and postdocs. I started my job right before the pandemic, and so they have helped me build a lab and a research program during these incredibly stressful and hard times. And so I need to thank my postdocs who lead the group, Dr. Gong Fan, Marjan Zamani, Thomas Gill, and Steph Allison Logan, as well as our incredible visiting postdoc, Graciela Cedeno. I need to thank my amazing graduate students, Alex, Cindy, Barath, and Nadia. And none of this work would be possible without the amazing MIT undergraduates. We have consistently had significant participation from undergraduate researchers that have driven our research program, and we are very excited to have them in the group, and we're looking forward to what they do next. So my email is on the top of the slide again. I really thank you for your attention, and I apologize again for not being able to be there in person. And I look forward to hearing from you if you have any questions about this talk. Thank you again to the organizers, and I look forward to meeting everyone in person in the future.